So the one question I kind of want to go over a little bit is the last question for the lab 8-9, and kind of go over the answers on this, because a lot of people kind of danced around the right answers but weren't necessarily saying things directly. So it's kind of important to talk about that a little bit. So the first part, what was the question being asked? What was the experiment testing? Okay, and that was all in the introduction of the lab, right? It talked about we wanted to kind of replicate some of those historical experiments to confirm that DNA was the genetic material. So I was hoping that your answer would involve that in some form or another. Some people mentioned transforming E. coli, that's good. Transforming E. coli from being kanamycin sensitive to kanamycin resistant, that is good. So all those things kind of combine together, make the right answer. What were the variables then, right? The independent variable, that is what the scientist is manipulating. So it's very simple. Did we add DNA to the tube to transform the culture, or did we not add DNA? So that was the independent variable. The dependent variable was the growth. Does it grow on kanamycin, or is it kanamycin resistant, or something like that? So in this answer, you had to tell me what was going on with 4 plates, right? So one of the plates was the actual experiment. Plate number 2 was where we added DNA to the competent cells and it became kanamycin resistant. All the other 3 are different types of controls. Plate 1 is the control for the independent variable, so there was no DNA added. The plate with no kanamycin was a control plate showing that the E. coli, in fact, was viable. It was able to grow, and therefore was kanamycin sensitive. And then the control culture was a positive control. This was the culture that was used in lab 8 to extract the plasmid from, and therefore it showed that we can have E. coli that is kanamycin resistant. So some people did a little better on this. Some people had a hard time articulating themselves, and that's part of the class, unfortunately. So you'll have to make sure you can articulate yourself for the lab exam. So let's talk about that lab exam. That's going to be this Thursday, and you're going to receive it in your email. It's going to be either a Word document or a fillable PDF document. I haven't decided yet what format it's going to look like. And most of the answers you can answer directly into the document, and then you're going to return that by submitting it on Moodle. Some of the questions may involve you to do some manipulation, so I may ask you to draw a graph, or I may ask you to draw a scientific diagram or something like that. And so you may need to print off a page or use a blank piece of paper or graph paper or anything like that. And that will have to be scanned, or you'll take a photo of it, and that will also be submitted on Moodle by 4 o'clock. Somebody's asking a question, can we use anything that is ours? Yes, you may. You can use the internet, you can use your lab reports, the lab manual, those kind of things. I do expect you to do the work yourself though, and I'll be looking very carefully to see, compare answers between students in the class. So make sure that you do the work yourself. And then at the end of that, you're going to submit it on Moodle, and then I'll mark it. So I think I said in the email what kind of things to expect. You're definitely going to do a lot of the same types of exercises we've seen already. So you begin images that you'll have to measure the size of a specimen. Remember, there were 2 ways to measure a size. There was proportion of field and using an ocular micrometer. Calculating magnification, so how big is a drawing, and those kind of things. And one thing that I usually get people to do in person is we do a standard curve exercise. And so with the email, I sent you the exercise that I usually provide. So it involves pipetting and those kind of things. Obviously, we're not doing the practical part of it, but you can do the data and manipulation. So you're going to receive a similar exercise where you're going to have some data. You may have to make a standard curve, and then determine the concentration of an unknown. So if you can't remember how to do that, go back and review what was done in lab 3, and definitely take a look at the exercise that I sent in the email because, like I said, I'm going to come up with something very, very similar. So you need to know how to do those calculations. Someone is asking, do we have to be able to use both methods to calculate size or just one? You should probably know both. It's handy, and that way, because I may ask you to do one or the other, or it may be easier to do one or the other for a particular image. Some of the questions will be kind of sort of traditional theory kind of questions, so they might be similar to the discussion questions asking you, how does this work, or how does that technique work, or something like that. So if you've been doing the labs all semester and you have a good grasp on them, you'll probably do well. If you have some areas that you're still struggling with understanding, now's the time to figure it out or ask me questions so that you can do well on the lab exam. So I think it's worth about 10%. It could be 12% of your grade. And so, like I said, if you've been doing well on the labs, that's going to be helpful because you don't know what you're doing. It's just a matter of getting it done in the allotted time. Okay, so that's all I have to say about the lab exam until Thursday. And unless anyone has any questions, just send them my way, please. So this is where we finished off on Thursday. I wanted to do one of these DNA transcription translation kind of questions and throw in a few mutation kind of questions around it. So this is kind of what the question is going to look like. Like I said, it'll be kind of a 5-mark question, and you'll have to do the transcription translation parts first. You may have to do things like label the promoter or the terminator. Do take a look at the problem I have put on Moodle with the exam study guide, and it gives you an idea of what it might look like. Obviously, I have to convert it to being an online kind of thing, but you'll have a genetic code provided for you, and so you don't need to like memorize all 64 codons. So here's the question. It's asking you, look at these particular places on the gene. What is going to happen? So the very first thing we need to do is we need to deal with the transcription and translation parts of this question. Okay, we need to figure out what does the gene look like and what amino acids are we producing here. So first thing you need to do, as usual, is always find that start codon. So there's the start codon. Don't get tricked by this one over here. As I mentioned before, that is not a start codon. It may look like it, you're thinking ATG, but that is reading backwards, right? Because for DNA, we always read 5' to 3'. So this is actually GTA, not ATG. So find your start codon. Here's a tip. I will always put the start codon in this class on the top strand. So hopefully that will help you and make it simpler for you, but it doesn't have to necessarily be the case. I just don't feel like tricking anybody this semester. I just want you to be able to find it easily. So find your start codon, and then you're going to make your messenger RNA. And like I said, the messenger RNA is going to look like the sense strand, and that's where the start codon is. So I'm just going to copy, and I'm going to turn all of my T's into U's. So this T here, of course, is turned to a U. This A is an A, and a C, and so on. So hopefully that's pretty straightforward. Then you find your start codon, and you underline all your triplets, or you identify all your triplets. And then what you need to do is basically take a look at the triplets, look on the genetic code, use those codons to figure out which amino acids you're going to code for. So here's our protein. Our protein starts at the start of codon, of course. The AUG codes for methionine. UAU codes for tyrosine. CCU for proline. And UAA is the stop codon, so that's going to be my little peptide there. So I do have a video, I think it was from last winter, that has these problems shown on it. I think it's all the same ones that were done in class, but I'm going to include that on the playlist so that people can take a look at that. But do, like I said, practice this so that you know what you're doing, and you can get those 5 marks on the final exam. So the next part of the question, it says, what would happen if the T at position 1 was changed to a C? Okay, So in this case here, I need to basically take that DNA and I'm going to change it to a TAC instead of a TAT. So notice I'm just looking at that one codon, okay? Because that's really what's important. So I need to know where that C is in the codon so that I can figure out what is going on here. So I have a TAC. Of course, the new RNA is going to be changed to a UAC. And then I can look that up on my genetic code. So UAC, and UAC codes for tyrosine. So I'm going from a tyrosine to a tyrosine, and this would of course be a silent mutation. So this would be worth 1 mark. You get 1 mark for kind of doing this part, showing me that you're getting a tyrosine, or sorry, 1/2 mark for showing me you get a tyrosine, 1/2 mark for telling me what type of mutation that is. And as a name, it's a silent mutation. So what was the second question? Second question says, what would happen if the G at position 2 was changed to an A? Now this is on the other strand, right? So you have to think a little bit about what is going on here. And this is, it looks like the 3rd codon of the gene. So the new DNA, I'm changing that GGA, so right here, to a GAA. And that would mean that the sense strand is now a CTT instead of a CCT. So figure out what my messenger RNA is. It's going to be CUU. Look that up on the genetic code, and we get an LEU, which is leucine. So we're changing from a proline to a leucine. So what type of mutation is that? That is a missense mutation. So like I said, 1/2 mark for telling me that you're changing your amino acid to a leucine, and 1/2 mark for telling me that you have a missense mutation. All right, so the 3rd one, let's see what this one says. What if the T at position 1 was changed to a G? Okay, so we have a TAT, and our new DNA is going to be a TAG, and so that to me rings a bell because I know that codon, and the new RNA is UAG, and the new amino acid is a stop codon. So there's no new amino acid. We're going from tyrosine to a stop codon. So this also has a name. This is called a nonsense mutation. So again, 1/2 mark for saying that we have a stop codon and that translation would stop there, and 1/2 mark for giving me the proper terminology, which is a nonsense mutation. Okay, and the last one, it says, what if the C at position 3 was changed to a T? Okay, so take a look at this. Because here's my start codon, right? And I've got these triplets. And remember that this one here is the stop codon, okay? So my mutation is on the other side of the stop codon. So what would happen to the gene? Well, the answer is not much. It's not within the coding part of the gene. So it's not going to affect the protein. It's not going to affect the amino acid sequence at all. It may affect some sort of regulatory component, like because the terminator is there, but that's something that we're not going to address in this course. So in this case, you would say the mutation is not within the gene, so the amino acid sequence is not going to be affected. Same thing if I asked about something that happens before the start codon. It might affect the promoter, might affect some sort of a regulatory component, but it's not going to affect the protein sequence. So you can say something like that. Okay, so hopefully these are pretty straightforward. I think we've done 3, maybe 4 of these now, and I've added a little bit of layers of complexity each time. So like I said, it'll be at least 5 marks on your final. I may ask multiple choice questions around some of these concepts as well. So know all those terms, the missense and the nonsense, and what those terms mean for those kind of mutations, silent mutations as well. And know how to do this kind of thing, because like I said, I'll be giving you a sequence of DNA to transcribe and translate for me. Okay, so 6 types of RNA. I keep reminding you, don't forget all your types of RNA. This unit, we talked about ribosomal RNA, transfer RNA, and messenger RNA, and sometimes we also talk about pre-messenger RNA. Remember, pre-messenger RNA is before it gets modified, before you get the splicing and the capping and tailing and those kind of things going on. Okay, so let's move on here. We're going to try to play a little bit of catch up, and I gotta just see here. Stop sharing that one and get to the next topic, the one I wanted to cover today. There it is. I guess I got to share it now. There we go. So topic 18 is about gene regulation. So these notes that you have for this unit are longer and more complex than the level I need you to know. I'm going to keep this short, and I'm going to skip over a lot of things. There's a lot of things in your notes that aren't going to be on the exam, and I'm going to try to point that out as we go along. But some of these things we've talked about, and some of these things are might be a little bit new. One of the key things is we have one more type of RNA to talk about, which we're going to get to. But I do want to kind of cover some of the basics around gene regulation. So the first thing to think about in terms of gene regulation is this little exercise here. So you can see I have a little sentence, and it says, add the punctuation. A woman without her man is nothing. So everyone's looking at this and thinking, well, of course I know how to punctuate this, right? So the men here are probably thinking, well, yeah, a woman without her man is nothing. And the women are like, whoa, wait. We're not going to punctuate it that way. We're going to punctuate it like this. A woman without her man is nothing. So you can see this is a really good analogy of gene expression. We have 20,000 human genes. We have all sorts of different cell types. And it's the same DNA in all our cells, but different things are going on with the same genetic material. So gene expression is kind of like this, right? We need to figure out where to put the pauses, when to have the breaks, and you're going to result in different kind of phenotypes in each of your different cells, right? So, you know, why regulate genes? So if you think about all these things, right? Sometimes our cells need to respond to environmental changes. You know, something, some sort of stimulus is coming along, and a gene is being activated that says, okay, we need to heal this wound, or we need to cool things off and sweat. It's too hot here, and all those kind of things. I guess I just said that in response to stimulus. Sometimes, you know, we're turning on one gene in one tissue. Our blood cells, they need hemoglobin. We don't need that in our skin. We actually have something kind of similar called myoglobin in our muscle tissues, but it needs to be turned on in the right places and at the right time. Some are turned on more often than others. Some are on always. Some are turned on only when needed. So there's lots of reasons for why we need to regulate genes. And how are we going to do this? Like I said, this can be considered immensely complex. You think about all of the things that we've talked about, right from the DNA level to the transcription and the translation, all those little steps in between. There are regulatory components to each of them. We want to conserve our resources. We don't want to lose anything. And we want to make sure everything is tight and perfect because, of course, you know, it could mean death for the cell or disease or something like that. So like I said, I kind of just want to touch on a few things today. Some that we've talked about already, and some that might be a little bit new for you, and mostly just kind of give you a taste of what gene regulation looks like. So if you remember, one thing to talk about is bacteria, which are quite simple in some ways. They don't have any compartmentalization. They have just a cytoplasm where all these processes are occurring, so transcription and translation. And remember last day we talked about how these things can actually happen simultaneously. So you can see we have a strand of DNA right here. This is my DNA. And you've got the RNA being produced. There's an RNA polymerase. And you've got, before the RNA is even done, we've got all these ribosomes that are binding to it and making proteins. So if you want to do something in a bacteria, you've got to be extra efficient. You don't want to waste all those ribosomes and all that ATP and start making a protein unless we really need to. So one thing that we did talk about way back, I think this was the topic on enzymes, and we talked about this in glycolysis, is that you can regulate the enzymes. So if you take a look at this pathway here, This is a pathway to make tryptophan, and there's a bunch of enzymes in this pathway. So you can see 1, 2, and 3, and you've got some sort of precursor, and it's going to get converted by an enzyme, and then you're going to have another step, and so on. You can see we've made tryptophan, right? So we talked a little bit about feedback inhibition, where basically this end product here, so the tryptophan, basically goes and inhibits one of the earlier enzymes. So most people are relatively familiar with this. It's a concept that's visited in high school. And but what we want to talk about is let's not even make these enzymes, because that takes energy to make enzymes. So let's regulate things at the gene level. So it's not that different in some ways in that an end product is going to regulate an early product. So this says here that cells can inhibit transcription. So remember, transcription is the RNA polymerase. So we can actually regulate the RNA polymerase right at the genetic level. So let's talk about how that is going to work. So there's our regulation of gene expression. In the same way, like I said, we can also use end products here as well. So let's talk about, we're going to talk about something called the tryptophan operon, okay? So here's some more genetics type words for you, right, that you're going to have to learn. And so I'm not going to ask you a big long answer question on an operon on the final exam, but I could imagine a couple of multiple choice. So it's important to know what one is. So you can see here's my definition of an operon, or part of a definition. It says a basic unit of genetic expression in bacteria. So how does an operon work? Well, an operon is the whole idea of multitasking. So rather than regulating one gene at a time, we would need 40,000 genes to do that, because every gene would have to have one regulatory component, right? So that's complex. So why not just regulate things, simultaneously? So you can see here what we have are basically 3 genes in a row, and we're going to make one messenger RNA. So you can make all 3 genes, or you can shut all 3 genes off. And so how are we going to do that? We're going to regulate this RNA polymerase. And you can see we're going to do this at the promoter. So the promoter has a region called the operator, where the regulation is going to occur. So I know I just threw a whole bunch of words at you. This is the overview. Let's take a look at this in detail, and I'll try to remind you of what some of these things are as we go along. So the operator is the first thing to note. Is this is a chunk of DNA. Okay, so this is a sequence of DNA, and it's found just before the genes, and it's a regulatory component. So you can see that the operator can get bound by certain proteins. Some of them are called repressors, and these repressors basically block the RNA polymerase. So the RNA polymerase can't do anything, right? We can't make any RNA if it can't bind DNA. So that's what an operator is. So let's take a look at an example. This is the tryptophan synthesis operon. So tryptophan is TRP. That's the abbreviation for tryptophan, if you see that. So like I said, you've got a whole bunch of genes here, 5 in fact, and they're all given names, the TRPA, TRPB, TRPC, TRPB, and TRPE. And you can see that all of these genes are regulated by the same promoter. And there's that operator. Like I said, it's a chunk on the promoter where some sort of regulatory protein can bind. So the default position for this operon is on. So what do I mean by that? I mean that these genes are just on all the time, because of course a cell wants to make tryptophan. It's an important amino acid. So you can see, just like a normal transcription, we have an RNA polymerase right there, and basically it's going to go along and it's going to make that messenger RNA, and the messenger RNA is going to code for 5 genes. So each gene has its own start codon and stop codon. You can see you're just represented by the little green and red bars. And then, of course, some ribosomes are going to find that long chunk of RNA, and the ribosomes are going to make all these enzyme subunits. So in this case here, we've got 5. So 5 genes, we're going to multitask 5 genes at once. So there's not much new here than what we've talked about, except for that all the genes are stapled together in one messenger RNA at this point. No regulation is occurring. Like I said, the default is this is on. The genes are just being done, transcribed all the time, until there's some tryptophan. and then that's where things are going to get regulated. So one other thing to note is there's one other gene to mention, is that somewhere else in the genome, you've got this regulatory gene. So you can see it's given a name, TRPR. And this here is going to make its own RNA. So this is messenger RNA here. I think there's a label for it. Here we go. Little chunk of RNA. And that little RNA is going to find a ribosome, and it's going to make a protein. And so the protein is going to, it's called the repressor. Okay, and I'll show you how that works in a moment. But basically, it's a little protein, and it's going to regulate this whole thing somehow. So like I said, there's the default position. We're not even thinking about that regulatory component. We're just thinking, okay, we've got these genes. Let's do something about it. We're going to put that RNA polymerase on there, and we're going to run with this. We'll look at the DNA. We'll make our RNA, and that RNA will find some proteins. Proteins fold into enzymes, and the enzymes make tryptophan. Okay, so we're going to make lots and lots of tryptophan. There's our amino acid. Of course, there's going to be a point where the cell has enough tryptophan. So this is where we have to start thinking about turning it off. We don't want to just waste all those resources, make those enzymes, and make tons and tons of tryptophan. So we want to turn this off at some point. So that's where that little regulatory component comes in. So there's that little regulatory component. Like I said, this is somewhere else in the genome, and it's just a gene, and it's being made into RNA by RNA polymerase. And then a ribosome is making this little protein. And this little protein, you can see, here's a note, it has an allosteric site that binds tryptophan. So this goes way back to our enzyme unit where we talked about allosteric enzymes, and we had said that basically what that means is they have a region on them that can bind some sort of regulatory molecule. In this case, the regulatory molecule is tryptophan. So there's the repressor. And you can see the repressor, this is the repressor here, this kind of big model of it. And it binds tryptophan. And when it binds tryptophan, the shape of this thing changes, and now it can bind DNA. So if you take a look, so there's the on position. I showed you this already a few times. Here we go. We're going to see what happens when there's too much tryptophan. When there's lots of tryptophan, here it comes. It's going to bind that repressor. The repressor changes shape, and it binds up there. So there's no label here, but this is the operator. And this is the RNA polymerase. So the RNA polymerase can't bind, so no RNA is going to be made. So that is how tryptophan works. Tryptophan operon works. Like I said, I am not going to ask you a big long answer question on the tryptophan operon, but what you should know is some of the basics, what an operon is, okay? And you know, the fact that it can turn on and turn off several genes all at once. And you might want to know a few of the terms around there, like operator, a couple of definitions like that might be handy for the final exam. Okay, let's move on. I'm going to leave this for now. I'm probably going to come back to this for our review class when we get to the end of the term here. Okay, so that's kind of what happens in bacteria. There are other things that go on in bacteria, but the main level of regulation is during transcription. Like I said, in bacteria, we want to do things super efficiently, so you do things right at the beginning. Transcription is at the beginning. In eukaryotes, it's a little bit more complex. We've got a nucleus, we've got introns, exons, we've got a lot more DNA. There's different cellular components. There's a whole bunch of things going on. And you can see I've got this list here of some of the things I'm going to talk about. If you go to the textbook, it is very long and very thorough. We're just going to cover some select examples here of things going on, and I'll try to highlight the things that are most important to know for Biology 107. Okay, so let's start right at the DNA level. Okay. Oh, and I'm gonna talk about this number 4 here in the most amount of detail, I think, today. So what's going on at the DNA level? There's a whole bunch of things you can do to your DNA. It turns out your cells can methylate nitrogenous bases. But one of the big things that they do is they can pack or unpack the chromatin. So if you take a look, we're talking about 2 types of chromatin now. So I mentioned before that chromatin, that word chromatin, basically means DNA and associated proteins, right? So which proteins are we talking about? Well, we're talking about those histone proteins and a few other proteins that are in there involved in the DNA packaging. So euchromatin, so U is the same U for eukaryote, right? So remember, eukaryote means true nucleus. So euchromatin means true chromatin. So this is kind of loosely packed, and since it's loosely packed, DNA and RNA enzymes, they can get in there and they can read the genes. So these are genetically active. Heterochromatin means it's packed and not active for those enzymes. So in different cells, we have different chromosomes and different portions of chromosomes open up or closed. And this is a regulatory a way to regulate genes entirely. If you have a calico cat, if you don't know what a calico cat is, Google that. It has all sorts of different interesting colors. And it turns out that some cells have some colors turned on and some cells have other colors turned on, which kind of gives them the interesting modeling pattern. So DNA level, number of things we can do. We can turn things on and off. Like I said, one way is just how we pack the DNA. So it's just like your house. Sometimes you have things accessible, and sometimes you have things packed away and hidden in the closet. And the things that are accessible are going to get used a bit more often than the things that are packed away for later. So here's an image, and it says, can you identify the following? And it's asking us to identify a bunch of things, the nucleus, the mitochondria, but also euchromatin and heterochromatin. So take a look at the image and see if you can find some of these things. Hopefully the nucleus is super obvious. It's this big thing here right in the middle. I'll get back to the chromatin in a minute. Let's see if we can find some mitochondria. I think this looks like a mitochondria right here. This is probably another mitochondrion right there. And maybe this one here and a couple of those other blobby things. Rough ER, so the rough ER you can see is all over the place here, all these little compartments with all those little studs, which are the ribosomes. The Golgi, I saw the Golgi here earlier, and I'm wondering if this is the Golgi. I'm seeing some blebs kind of come off of it. So what about chromatin? So this stuff here is loose, right? So this is the euchromatin, and this stuff here is condensed. So actually that one there might be a nucleolus. It's kind of hard to tell, but that's kind of what we're looking at when you see these images. I think I actually have the labeled one here. And so yeah, they're saying that that's the nucleolus there, but this is the heterochromatin. Like I said, it's kind of condensed, and therefore it stains a lot darker under an electron micrograph. And I was right, there's the Golgi. Like I said, it's usually pretty small, but you can often identify it by the little vesicles that are kind of budding off of it. So we talked about these transcriptional controls with prokaryotes, and it turns out there's a ton that happen in eukaryotes. I'm going to mostly skip this section. I just want to show you one image, and there's like a whole page of notes I've given you. Like I said, we're going to skip over it, but it involves a whole bunch of things. Right? It involves the promoters, regulation there. There's repressors, enhancers, and silencers, and transcription factors. That's kind of the main word that I want you to know, is transcription factor. And that's something we talked about last day. If you don't remember what a transcription factor is, you probably want to know what that is for the final exam. So I'm going to skip through this, like I said, really quick. You can see it's showing all these different things going on, but I don't really want to talk about it for this, this semester. We're just kind of running out of time, and there's more important things to discuss. RNA processing, we talked about the things that are going on, the splicing, and sometimes there's regulation that goes on there. Some RNA molecules are more stable than others. So if an RNA molecule is very stable, it's going to sit around for a while, and the genes and the gene products are going to be made off of that RNA. Some are digested very quickly, and so that's another way to regulate your RNA. You can put a long tail on it or a short tail on it, and that will help it to be stable. So there's alternate splicing. I mentioned that this was important in the production of antibodies for your immune system. So that's something to remember what that is. So there's a whole bunch of translational controls. So by that, I mean things that are going on at the ribosome. Again, something I'm not going to talk about. What we do want to talk about is what happens after translation. And I want to touch on a few of these things. Notice E is in bold. That's the one we're going to talk about the most. But let's talk about a couple of these here. And so one of them is formation of complexes. So a number of proteins, by the way, are not just one polypeptide strand. A lot of proteins like this one here, this is ATP synthase. It's something like 15 different proteins, and this thing is not active until it's in the right place and assembled with all its subcomponents. So this is important, right? They gotta go to the right location and they gotta assemble properly, and that is what's going to be part of the regulation. A lot of proteins are modified in all sorts of different ways. Some of them, you can see here, some things that are listed here. So proteolysis means they're cut up. Glycosylation, so I'll talk about that in a moment. Some are phosphorylated, that means we're adding a phosphate group to them. Some are lipidated, that means you add a lipid to them. And we're going to talk about this in detail, but targeting to specific cellular locations. So what was glycosylation? That is where we're adding carbohydrates. So remember, glycosylation happens in ER, and it also happens to some degree in the Golgi body. And so what are we looking at here? We're looking at some carbohydrates. So these are the carbohydrates that are found in the blood groups. So if you have the A blood group, you have this carbohydrate here. So this is, I think that's N-acetylgalactose. And if you have B type blood, you have galactose, not the N-acetylgalactose. And that's enough of a difference that if you are A type blood, you can't take a transfusion from somebody with B type blood. If you're B type blood, you can't take a transfusion from someone who is A type blood. If you have O type blood, you don't have that antigen at all. So you have a third type of blood, right? Not going to get into all the biology of blood groups, but just pointing out that these are carbohydrates, and that's what glycosylation means. And these things are not active until the carbohydrate is added. So there's our rough ER, and there's our Golgi, and that's where the glycosylation occurs. Okay, so this is what I wanted to talk about today in a little bit of detail, is targeting things to specific locations. So we're mostly going to be talking about targeting things to the endomembrane system or targeting things to be exported. So by exported, I mean secreted from a cell. So here's an example here, right? This is insulin, and insulin is made by your pancreas cells, and it gets secreted into your blood after you eat some sugar. So you can see it says a note here. It says here, this has something called a signal peptide at its N-terminus. So let's talk about what that means. I'll come back to that in a second. So a signal peptide, it says here, is a short amino acid sequence, and it's presented at the N-terminus. So that means the beginning of a protein. And so this is kind of what it looks like, right? You've got the beginning of the protein, so it's called the N-terminus because it's got a nitrogen amino group there. And the other end is called the C-terminus. So signal peptide is a specific amino acid sequence found at the beginning. So it's kind of the start of something. It's kind of like, I mean, if you read a book or a novel, often it has a table of contents, and the table of contents is the beginning, and the table of contents tells you what to expect. So same thing with this protein. It's got the first few amino acids are like that table of contents. They tell you what to expect, where this protein is going to. And so this is called a signal peptide. That's the name that scientists have given it. And this is a region, you can see it has some a hydrophobic region. It's got a lysine in there and a few other characteristics. If you want, you can always look that up and see what it says. And so this basically says, send me to the ER. That's what a signal peptide is, right? It's an amino acid sequence that says, hey, I want to go to the endoplasmic reticulum. So there's just a little bit of information about it. It's got some positively charged amino acids. I said like lysine has some hydrophobic ones, and then some uncharged ones, and then there's a cleavage site. And then there is the amino acids for the actual protein, the functional parts. So I told you I want to go back. I was just going to go back to this. This is slightly out of sequence. This is just a reminder that, remember, we have two types of ribosomes. We have the free ones that are found in the cytoplasm, and we have the bound ones, the ones that are going to the ER. And these bound ribosomes are making proteins that are targeted towards a whole bunch of things, the whole basically endomembrane system. So that includes the Golgi, vacuoles, peroxisomes, lysosomes, and anything secreted. Okay. So here is our definition of a signal peptide, and let's take a look how this process occurs. So unfortunately, this is a very busy image from the textbook, and so I'm going to try to go through this step by step. So if you take a look, there's our ribosome, and our ribosome has a little bit of messenger RNA. I don't know if this is labeled or not, but I can write on this is my messenger RNA right here. And so all messenger RNAs, they find the ribosome and it all starts off in the cytoplasm. That's where translation begins. So you can see what is happening is that there's the messenger RNA, there's a little label. The first few amino acids, right? So I think it's about 20 or 30 amino acids. Like I said, are kind of like the table of contents. They're the signal peptide, and they say, hey, bring me over to the ER. So there's this little thing here called the SRP. So the SRP stands for signal recognition particle. Okay, not protein, but particle, because it's not just made out of protein. So this is actually a protein RNA complex. It's made out of protein and RNA. So our last type of RNA we're going to talk about here. So this is something that binds the signal peptide. So it's a signal recognition particle. So it's going to recognize a signal. And what's that signal? The signal is the signal peptide. Okay, so hopefully you're still with me here. We've got translation is occurring as usual. A few amino acids are made, and sometimes those amino acids have a certain chemical signature. They're hydrophobic with some positive charges, and that is a signal to the cell to say, hey, come grab me and bring me over to the ER. So there's the ER there. And this is going to be delivered to the endoplasmic reticulum. So the endoplasmic reticulum has receptors on it, and those receptors are designed to bind the SRP, because the SRP is going to bind that ribosome and is going to bring it over to the ER. So there's the receptor. It's grabbing onto the ribosome and the protein that has started translation. And I guess we've said all that already. The signal peptide interacts with the signal recognition particle and is moved to the ER, and that sometimes is called translocation. That means moving from one place to another, translocation. So after that, it's going to dock on that membrane, and there it goes. It's docking, and translation is going to resume. So it actually pauses translation. It says, just stop for a second. We're going to bring you over to the ER, and then you can finish your job. So over at the ER, we're stringing on more and more amino acids. And there we go. And you can see that the protein itself is going to go through a little channel, and the protein is going to be produced, and ultimately it's going to be made inside the ER. So there it goes, and when translation is done, remember it gets to the stop codon and everything disassembles, but the ribosome stays out in the cytoplasm, and same thing with the messenger RNA, but the protein itself at this stage is now found inside the ER. So the inside of the ER is called the lumen. If you ever see that term, ER lumen, that's inside of the ER, and so the protein is there now. So I want to show you now what happens, okay? Because the protein is, it's in the ER, and how does that get exported? Like if this is, if this is insulin in a pancreas cell, how does it get out of the cell now? So remember that all these things are connected. So there is our endoplasmic reticulum, and this little green thing here, this little green thing right there, that's the protein. So it gets made inside the ER, it's folded, it's ready to go, and now it needs to go somewhere. So it ends up getting packaged into a vesicle. So there's a little vesicle forming, and right here, this is the vesicle. And so the vesicle is going to go, and it's going to now fuse with the Golgi. So remember, the first part of the Golgi is called the cis face of the Golgi. It's going to travel through the Golgi. I think that part of the mechanism is poorly understood. There's a few models that kind of explain it, but I don't think anyone's actually ever proven anything yet. And eventually ends up at the trans face of the Golgi. So cis means close. Think of it like sisters being close, right? And then trans means kind of opposite, like a transatlantic flight or something like that, right? And so now the protein is in a vesicle after being, going through the Golgi. It's probably getting glycosylated, maybe modified in another way. And then kind of the same thing happens. That vesicle fuses with the plasma membrane, and the protein itself has exited the cell right there. So it could be delivered to one of the other compartments of the endomembrane system. It could be delivered to a lysosome, or maybe back to the rough ER, and it's kind of a similar process. Sometimes they have extra bits of information in their sequence that allow them to do that, but we're not going to get into all that in this course. Just can only cover so much. Okay, so what's some of the takeaway messages here? Takeaway message is for bacteria, we have operons. And operons are where we multitask and we control the transcription of multiple genes at once. It could be 3, could be 5, could be 7. In the case of the tryptophan operon, we're looking at 5 genes all at once controlled at the transcriptional level. For eukaryotes, there are a whole bunch of things that can happen. There's all these stages, and there's regulation all the different stages. So a couple of things we touched on were, of course, DNA can be packaged differently. We have the euchromatin and heterochromatin. We have regulation that happens at transcription, translation, all the stages. And we talked a little bit about what happens post-translationally, right? What happens at the very end, it gets glycosylated and often targeted to certain organelles or being secreted. And the other take-home message is that now we've covered all the 6 types of RNA. So this slide here, take note, big star, another big star, right? As I mentioned, this slide, 100% for sure will be on the exam. Last year, I basically asked people to fill in the blanks. So I would have either the name of the RNA or the definition, and that's it. No tricks. You need to know this slide, 100% for sure. For some reason, people don't want to give me all the information. You can't just put tRNA. You got to spell out the whole word. I'm making an easy question. I haven't decided whether I'm going to be exactly the same this year or not. I mean, it could be matching, or I could have some other kind of modification kind of question. It's not going to be complex if you know all of these. So let's just review them quickly, and then we'll finish up today. So what is the messenger RNA? The messenger RNA carries the codons. So that would be the amino acid sequence. Transfer RNA binds to an amino acid, brings it to the ribosome, and matches up the appropriate anticodon to the codon on the messenger RNA. Ribosomal RNA has structural and catalytic roles in ribosomes. Telomerase RNA, that's used as a template by telomerase to extend the telomeres. Small nuclear RNA, structural and catalytic roles in spliceosomes. And lastly, signal recognition particle RNA. So this is a component of the signal recognition particle. And you actually don't need to know that other part. So all of these things, why am I asking you to know all these types of RNA? Because look at all this information packed into one slide. What is a ribosome? What's translation? What is a telomere? So all these concepts are important to know. So like I said, this is going to be 6 marks of your final exam. That's 6 marks out of 80. I'm telling you for sure, this is going to be on there somehow. Please go and learn it. I want this question to be easy for you. I think that is the end of today's lecture. I got a couple of things to say. We have 2 units left, and I'm hoping the next 2 units will be kind of fun and interesting for you. We're going to talk about biotechnology and forensics, and we're going to talk about viruses. So lots of really interesting information and some really cool things we're going to discuss over the next 2 to 3 lectures. And hopefully, if you've been paying attention, all these things here we've been talking about, this transcription and translation, these things are going to be important principles so that we can understand a little bit about genetic engineering, a little bit about viruses. And then after that, I'm hoping we'll have some review, and then that's the course. So hopefully it's all fun from now on. other than the lab exam, which is some people's, I guess, version of fun, and then the final exam. So there's a lot we've learned. I encourage you to, start looking over your notes and doing a little bit of studying now as the final exam is coming closer. I'm hoping people will have some good review questions for me to go over for the final exam when we get to the review part. So that is all for today. I know today's lecture was kind of complex, but I think the next ones will be very interesting, a little bit more enjoyable. So that's all for now. I will see you on Wednesday.